Hello friends, welcome to my course on Microbial Biotechnology. We are in module 3, currently discussing about the physiology of extremophiles and the adaptations. In today's lecture, we will be learning about the halophiles, xerophiles, radiophiles and metallophiles. So, let us start with the adaptability to variable saline or osmotic conditions. So, you can see in this picture a saline environment. So, salinity strongly influences microbial community ranging from marine environments to soda lakes and salt inclusions. Halophiles can tolerate high salinity levels by achieving osmotic balance through the accumulation of potassium ions or the synthesis of compatible organic solutes. Water activity can be regulated by the production of metabolites capable of storing or attracting water. Microbial life has been observed in brines with high concentration of chaotropic salts such as magnesium chloride and calcium chloride. Now, what are the effects of changing osmotic concentration on microorganisms? So, here is a isotonic solution, hypertonic solution and a hypotonic solution, and you can see here in the first case there is no net movement of water, cell membrane is attached to the cell wall. In a hypotonic solution, water particles move into the cell. As you can see, cell wall counteracts osmotic pressure to prevent swelling and lysis. In the hypertonic solution, water particles move out of the cell membrane shrinks. As you can see, the cell membrane is shrunken and detaches from the cell wall. So, this is called as plasmolysis. So, as food matrices become dehydrated, most microbes cannot survive. Brines and layering meat and fish in salt are the time tested methods for preserving food due to these reasons. What are the adaptability mechanisms employed by halophiles? They have evolved a range of adaptations that enable them to survive and thrive in high saline environments. For example, they deploy osmotic regulation, then salt resistant proteins, DNA and RNA stability helps in these adaptability. Then they have regulation of the ion transport and the membrane composition is also modified. There are thermophilic adaptations and of course, genetic adaptations, and in many cases there is biofilm formation. So, salt and pH homeostasis very important, which leads to the protein stabilization. And then here one thing that is observed, they have large halophiles and they also engage polyploidy. And salt in and salt out strategies are one of the important key factor which helps in adaptability. And then the presence of osmolytes and biosynthesis of organic solutes such as glycine, betaine, actoin, etc. helps in this kind of adaptability. So, there are here are some example. This is Salinibacter rubor. This is a red bacterium found in Saltern crystalliza pons in Alicante, Spain. And this is Wallemia. This is a found in hypersaline water of solar salterns, bitterns and salted meat. And this is a green algae, Dunella cellula. So, let us discuss these various strategies, osmotic regulation and others one by one. So, in osmotic regulation, halophiles use compatible solutes such as glycerol and trehalose to balance osmotic pressure, prevent dehydration and stabilize cellular structures. Additionally, some employ a salt-in strategy accumulating potassium ions within the cell to match the high salt concentration in their surroundings. Salt resistant proteins. The enzymes and proteins of halophiles are specially adapted to function in saline conditions. These proteins often feature a high proportion of amino acids and flexible structures that help maintain stability and activity despite the high salt content. DNA and RNA stability as one of the strategies are very important. These halophiles typically have high G+C content in their DNA, which contributes to greater stability under extreme conditions. They also produce protective proteins that shield both DNA and RNA from salt induced degradation. Then there are ion pumps and transport. There are specialized ion pumps such as the sodium hydrogen antiporters help regulate internal salt levels, while potassium transporters are crucial for maintaining osmotic balance by accumulating potassium ions inside the cells. The cell membrane composition to preserve the membrane integrity and fluidity in saline environments, halophiles adjust the composition of the cell membranes by increasing the proportion of unsaturated or branched chain fatty acids. Then there are certain thermophilic adaptations. Many halophiles are also thermophiles, meaning they can survive at high temperatures. Their proteins are adapted to function effectively in both high salt and high temperature conditions. Then there are certain genetic adaptations which arises from genetic diversity in halophiles, which enhance mechanisms like polyploidy, having multiple copies of the genome, and horizontal gene transfer, which allow them to rapidly adapt to changing conditions. Then biofilm formation. Some halophiles form biofilms which offer added protection against environmental stresses like high salinity and UV radiation. Let us now discuss about adaptability to variable water activity, or AW. Water activity indicates the amount of available water in a substance that can support biological functions with values ranging from 0 to 1. Water in food can be bound through solutes, ions, hydrophilic colloids, or water of crystallization, and microorganisms require available free water for growth. Most microorganisms grow well at water activities around 1 or 0.8. This is why drying food or adding large quantity of salt and sugar is effective at preventing food spoilage by lowering water activity and limiting microbial growth. So, xerophiles are extremophiles that thrive in environments with low water availability. They can grow at low water availability below 0.8 and are commonly found in dehydrated foods, saline lakes, deserts, and other areas with limited moistures. Xerophiles are similar to halophiles because both thrive in environments with low water scarcity or activity, with low water activity, not scarcity. Despite the importance of water for biological processes, xerophiles have evolved mechanisms to survive in conditions with limited water availability. So, whenever there is a dehydration due to maybe high salt or high sugar or any other conditions, so the cell will come under various stress like physical stress, physiological stress, and then metabolic stress and biochemical stress. So, here there is loss of in the physical stress there is loss of cytoplasmic volume and cell shrinkage, decrease in the target pressure and cell surface area. Then under physiological stress there is a loss of membrane integrity and leakage of intracellular components, disruption of the ion potential across the membrane, denaturation of membrane bound proteins, and under metabolic stress it will have inhibition of photosynthetic machinery, disruption of metabolite transport through the membrane, alteration to the kinetics of metabolic pathways, and rates of energy production. And it will also face certain biochemical stress where there is accumulation of ROS that damage macromolecules, denaturation of intracellular proteins, DNA oxidation, alkylation, and chemical modification. So, this is the impact of low water activity on microbial cells. So, how does a xerophile address this kind of stress or challenges? So, for these various mechanisms are there. For example, energy reduction, water loss prevention, increased water retention, DNA and protein protection, use of osmoprotectants, metabolic activity alteration, downregulation of photosynthesis, upregulation of ROS, scavengers and DNA repair proteins, accumulation of compatible solutes and salts, phospholipid modifications, formation of biofilms, and also sporulation. So, these figure these adaptation mechanisms overall. Now, there are another kind of extremophiles known as radiophiles. These are microorganisms which can adapt to high radiations. For example, Deinococcus radiodurans is highly resistant to gamma radiation due to its efficient DNA repair mechanisms. When exposed to radiation, it experiences severe DNA damage, but multiple copies of its chromosomal DNA facilitate repair and replacement of fragmented sections, maintaining cell viability. In addition to radiation, Deinococcus radiodurans is resistant to UV radiation, mutagenic chemicals, and desiccation. Its multigenomic structure with several copies of its genome enhances DNA repair and contributes to its exceptional resilience. Hypothermophilic archaea such as Thermococcus littoralis and Pyrococcus furiosus also survive high levels of gamma radiation. So, this is the overall radiation spectrum, and you can see the visible spectrum over here, and then you have the ionizing radiation with its short wavelength, which has high energy, and this is harmful to most microbes. UV radiation at around 260 nanometer is particularly damaging as it is strongly absorbed by DNA. Neo UV radiation between 325 to 400 nanometer can also harm microorganisms by breaking down tryptophan, leading to toxic photoproducts and DNA strand breaks. Sufficiently intense visible light can also damage or kill microbial cells. Photosensitizers including pigments like chlorophyll, bacteriochlorophyll, cytochromes and flavins absorb light energy and become activated. The activated photosensitizers transfer energy to molecular oxygen, generating singlet oxygen, which can be harmful. So, we see that radiation is a big challenge for the microbes to survive. So, there is some example here. Sulfolobolus tocadi increases expression of rad B and rad A in response to UV radiation, which is a survival strategy. Then you have dictyostelium, which uses translation synthesis, the Fanconi anemia pathway and nucleotide excision repair to survive radiation and DNA cross-linking agents. Then you have others like Chlamydomonas and then D radiodurans, which have been studied for their extremophilic traits such as extremolites, extremogymes, and unique proteins like histone, like DNA binding proteins HU, which aid in survival under radiation. How do microorganisms adapt to radiation? Lower levels of ionizing radiations cause mutations which renders lethality in the long run. Higher levels cause breakage of hydrogen bonds, oxidize double bonds, destroys ring structures and polymerizes some molecules. So, yeah, you can see the DNA double breaks occurring over here, but then there are repair mechanisms which restores these back. D radiodurans stands out prominently due to its remarkable ability to reconstruct its genome following extensive radiation exposure, a phenomenon that captivates microbiologists and sparks intense scientific curiosity. What are the resistance mechanisms of these D radiodurans? These form tightly linked genome structure under ionizing radiation to protect its DNA. It can repair up to 200 double strand breaks, whereas E. coli succumbs to as low as 12 double strand breaks. The bacterium accumulates the manganese complexes that counteract iron dependent ROS, preserving DNA repair enzymes. An alternative theory suggests that small symmetric antioxidant complexes protect the proteome from IR damages. Cross kingdom analysis supports the role of hydrogen and manganese complexes in cellular IR radiations. The combined effect of these mechanisms and radiodurans evolutionary adaptation empower its radiation resistance. Another type of extremophile is the metallophiles. As the name suggests, these microbes can adapt to high metallic metal ion concentrations in their habitat. So, the adaptation mechanisms for metallophiles include conversion. The metallophiles utilize enzymes to convert these metals into less toxic or less bioavailable forms, reducing the metal's harmful effect on the walls on the cell. Then efflux. active removal of metals from the cell through specialized efflux system or pumping systems. These adaptations allow metallophiles to survive and function in extreme metal rich environment. Now, let us also discuss some other adaptations apart from the ones studied, particularly adaptation to variable oxygen concentration. So, here you can see the various tubes, and you can see that there are two zones. On the top of the tube you have rich area which is rich in oxygen, and below you have area which is poor in oxygen. So, this is known as the anoxic zone, and this is the oxic zone in terms of oxygen content. So, if we grow certain bacteria, some will be growing in the top, some will be growing at the bottom, and some will be dispersed throughout with some concentration in the top, and some will be very, very uniform, and some will be not growing at the top, neither at the bottom, but just a little bit submerged. So, this is something very interesting phenomena. So, accordingly they have been classified as obligate aerobe, facultative anaerobe, aerotolerant anaerobe, strict anaerobe, and microaerophile. So, these obligate aerobe completely dependent on atmospheric oxygen for the growth. They cannot survive without oxygen. Facultative anaerobe do not require oxygen for growth, but grow better in its presence, and aerotolerant anaerobes grow equally well whether oxygen is present or not, but they can tolerate oxygen, but they do not make use of it. Then there are strict anaerobes. Oxygen is toxic to them, and they are usually killed by the presence of oxygen, and this is microaerophile, damaged by the normal atmospheric level of oxygen around roughly 20%, and require oxygen level within 2 to 10% for their growth. So, they will survive well under a little bit of submerged condition. What are the adaptation mechanisms to variable oxygen concentration? So, the toxic oxygen derivatives are formed when cellular proteins such as flavoproteins transfer electrons to oxygen. These oxygen derivatives are called reactive oxygen species, and they can damage proteins, lipids, and nucleic acids. So, we may have superoxide radical, then hydrogen peroxide, and hydroxyl radical. Microorganisms must be able to protect itself against the reactive oxygen species, but how do they do it? So, one of the mechanism is using the ROS to destroy invading pathogens. Neutrophils and macrophages, two important immune system cells, use ROS to destroy pathogens. So, here we call it as oxygen dependent killing. Many microbes use enzymes that can protect against toxic products of oxygen. Obligate aerobes and facultative anaerobes usually contain the enzyme superoxide dismutase and catalase, which catalyze the destruction of superoxide radical and hydrogen peroxide respectively. Peroxidase can also be used to destroy hydrogen peroxide. Strict anaerobes lack these enzymes or have them in very low concentrations, and therefore cannot tolerate oxygen. Severe oxidative stress or apoptosis occurs. During severe oxidative stress and DNA damage, hits of protein 60 indirectly plays a role in apoptosis induction. In response to oxidative stress, the SSP complex assists in localizing the FHIT protein to mitochondria, where it stabilizes ferrodoxin reductase, as you can see here. This leads to increased production of reactive oxygen species. Many of them are getting produced, triggering the release of cytochrome C and subsequent activation of caspase, ultimately leading to the apoptosis. In the case of genotoxic stress, FHIT functions as a part of the checkpoint response to DNA damage through CHK1, resulting in cell cycle arrest and potentially apoptosis of the DNA damage is severe. Now, there are certain genetic adaptations through horizontal gene transfer, mutations, and quorum sensing, which helps in this adaptation. So, horizontal gene transfer allows them to gain new traits via transformation, transduction, and conjugation. Mutations help develop resistance to antibiotics in species like E. coli and Microbacterium tuberculosis. And then in quorum sensing, which enables communications based on population density influencing behaviors such as bioluminescence in Vibrio fishery and virulence in Pseudomonas aeruginosa, enhancing their resilience in various environments. So, conjugation is a type of horizontal gene transfer which involves direct contact between a donor and recipient cell via filamentous protein SX pilus, pulling them together. The donor transfers all or part of its plasmid through the pilus to the recipient, as you can see in this picture. Then there is transduction in B, which involves a bacteriophage vector delivering genes between bacteria. When the phage attaches to a bacterial cell, its DNA gets incorporated into the host chromosome. During its replication, the phage may acquire bits of host DNA and carry them to new host. Bacteriophages can also acquire transposons, which are DNA pieces capable of relocating from one DNA to another and transfer them to new bacterial cells. Then finally, we have the transformation that involves uptake of naked DNA from the environment into a cell where it gets integrated. This process occurs randomly in natural settings as the DNA fragments originate from lysed cells. However, only competent cells in a specific physiological state permitting DNA uptake can incorporate these fragments. What are the microbial adaptations to enhance stress tolerance? Retention proteins in adaptation. Microorganisms use retention proteins, particularly transcription factors, to adapt to environmental changes by regulating gene expression. In Saccharomyces cerevisiae, repeated exposure to galactose increases the expression of galactose metabolizing genes, an effect that can persist even without galactose, influenced by transcription factors like GAL3 and GAL1. When microorganisms are subjected to environmental stress, the long retaining proteins are produced to protect the cell for a long time. So, you can see here these long retaining proteins and the general proteins present over here. Epigenetic modifications also helps in adaptation. Epigenetic changes, modifications to DNA that control gene activity without altering the DNA sequences, help microorganisms adapt to stress by temporarily altering gene expression. Though these changes are not passed to future generations, these modifications linked to short-term adaptations affect DNA histone interactions and chromatin structure influencing phenotype. For example, histone acetylation aids in repairing UV damaged DNA. In this picture, you see when organisms are stimulated by external environmental stress, epigenetic modifications are altered, affecting transcription and the translation process. Then there is also cross protection against stress. Cross protection basically refers to a phenomena where exposure to one pathogen or stressor protects another, often a related pathogen or stressor. Microorganisms can develop enhanced stress tolerance after adapting to a specific stressor, leading to improved resistance to other stresses. For example, Lactobacillus rhamnosus and Saccharomyces cerevisiae show increased tolerance to various stressors after adapting to one. The cross tolerance might involve gene environment interactions or common stress response mechanisms, though the exact process remains unclear. So, in this picture, we can see microorganisms are adapted to a stressful environment, resulting in cross protection against stress. Then quorum sensing is an important adaptation strategy in prokaryotes. Quorum sensing is a communication mechanism that allows prokaryotes, particularly bacteria, to sense their population density through signaling molecules called autoinducers. These process enables coordinated behavior that enhance survival and adaptability in various environments. For example, in Pseudomonas aeruginosa, it employs a quorum sensing mechanism to coordinate the production of virulence factors and biofilm formation, optimizing its pathogenic potential. So, in this case of quorum sensing, the LUX I makes a signal molecule called AHL, which moves easily in bacteria, while LUX R helps turn on specific genes when it binds to the AHL. So, this is one of the methods. Here the transcription is not activated at low cell density, transcription is activated at higher cell density. So, with this we come to end of this part of this lecture. Thank you for your patient hearing.